really exciting, I think, to be back in the parasitology world, where I lived for a long time with Trypanosoma brucei. But currently, indeed, I'm mostly focused on mammalian metabolism, and that's what I will discuss with you today with a digital twin approach. So, as I said, I'm mainly focused on metabolic diseases, and I'm in a pediatrics clinic in Groningen. So I'm not a medical doctor, for those who don't know me. I'm a biochemist, and I have been modeling since I was a master's student, but always in close relation to the laboratory. And since 2009, I'm based in a pediatric hospital in Groningen. So I collaborate a lot with the clinicians who work on these inherited metabolic disease, which are chronic and very debilitating diseases for children, often lethal. How I look at this as a systems biologist is that these inherited monogenetic diseases are actually forming a continuum with the multifactorial diseases. So, of course, in a single gene deletion, you have only one mutation, but there is a whole system around this that determines what will be the outcome in a certain patient. And there are, of course, today also a lot of previously non-metabolic diseases like cancer or Parkinson's, where we now are aware that metabolism is playing a key role. So in my view, this is really a continuum. But when we focus on the inherited metabolic diseases, I think this slide is totally outdated because they're not more than 600, but more than 1,800. They're often single enzyme deficiencies. Sometimes they are assembly defects in protein complexes or whole deficiencies of an entire organelle, like a peroxisome. Many of those affect children because they are so central in our cellular function. And the symptoms can range very widely, and also the severity from lethal to mild or late onset. And this may even be the case with the same mutation. So two siblings may have a very different type of symptoms and also disease severity. Typically, these diseases affect multiple organs. And when you treat them, of course, you would like to repair the gene. And this is upcoming, but this is not like standard of care at the moment. The standard of care would be to give a special diet, to supplement certain cofactors, or to avoid certain nutrients. So this is also systemic. The food is, of course, affecting the whole body, all the cells in the body, and the whole metabolic network. So that's why I think this is really systems biology, and I hope to convince you of this. In Groningen, the University Medical Center is expert center for a couple of these inherited diseases, mainly glycogen storage diseases, the medium chain and multiple acyl-CoA dehydrogenase deficiency, and that's also what I'll talk to you about, and a number of amino acid related deficiencies. So if we talk about the fatty acid oxidation, this is a mitochondrial pathway. So I will go a bit in depth into the biochemistry because we need to model for certain applications, of course. This is mitochondrial. It's also peroxisomal, but I'll focus on the mitochondria. This is a rare disease, but among the fatty acid oxidation disorders, not the most prevalent one. In the Netherlands, 1 in 8,000 newborns, we screen for it. The children are apparently healthy, and that's also the risk of it because you don't see it on them. But there is a very high risk in these children of hypoglycemia, so a very low blood sugar level, and this could even lead to sudden infant death. And that can happen like overnight when the child may have a little bit of a fever, is not eating well, so fasting longer than normally, and then suddenly these symptoms appear. Well, you can avoid this, of course, with regular feeding, and particularly when the child has a fever, you can be very cautious, but it shows very clearly that there is a loss of metabolic robustness in these children. And what we also see here is a lot of patient-to-patient variability. So even siblings with the same mutation could have a very different outcome. And if I look at this with a systems biology or systems medicine vision, I mean, we all have, of course, our own genetic makeup. And even if you have the same mutation, then the rest of the genome could be different between siblings. They have their own history, lifestyle. So this all integrates to develop disease symptoms, but also response to treatment and the whole prognosis. So what we like to do is use all kind of omics analysis and computational model to get, first of all, a better understanding, and then hopefully also give better advice to the clinicians and to the patients. So a bit of biochemistry. Fatty acid oxidation is a pathway where we break down fatty acids, and these are very long chains of carbons, and they will be linked to a coenzyme A. And remember the coenzyme A, because this will be the main character in my talk. And what happens in the fatty acid oxidation is essentially that these acyl chains, these long carbon chains, they are shortened, and then you chop off 2 carbon atoms in every round, and you have acetyl-CoA, and this is then further oxidized to CO2. But I'll focus on this process of shortening the fatty acids. And if we then look at the pathway with a bit more of a systems view, then what we see is that this is a cyclic pathway, because we enter a fatty acid, so palmitoyl would be 16 carbon atoms. It enters into the mitochondria, so we have here a C16 acyl-CoA molecule. It goes through a number of reactions, and then in the end you end up with another acyl-CoA, but it's just 2 carbon atoms shorter, because you chop off this acetyl-CoA. So you have to go through these reactions multiple times. So it's cyclic until you completely break down the whole long chain into pieces of 2 carbon atoms. And in fact, there are inherited errors of metabolism associated with all of these enzymes, or almost all of them, but this MCAT deficiency is the most often seen. Well, why is this leading to hypoglycemia? Because I said low blood glucose is a symptom. Well, the reason is glucose we can get from glycogen during fasting, but we can also make it from amino acids or from lactate or from glycerol, but that requires ATP. And the ATP, of course, you're not getting from glucose. That would be a waste. That would be a futile cycle. So you need to get that from fatty acid oxidation. And that is why these fatty acid oxidation disorders can lead to hypoglycemia. Well, if we now look at the pathway, we're going to model it in this talk. So what's a very important principle, in my view, is that enzymes, they communicate via metabolites. So in all our models, what is a central idea is that an enzyme will become faster when there is more substrate, or slower when there is more product. And this is what we have to capture in our equations to predict what will happen if we have a mutation in one enzyme, what will happen to the rest of the pathway. So assume we have a mutation in this enzyme number 2, meaning that the activity is less, then there will be less of the product of this enzyme. And the other enzymes will only know about this because they have less of a substrate. And then the next enzyme, of course, also has less of a substrate. And this is how this kind of goes as a wave through the whole system. So this is how we model systems, but also how they behave. All these local interactions, they govern the behavior of very large and complex networks. Well, one of these very important molecules is coenzyme A. Coenzyme A, you saw already, is involved in the fatty acid oxidation, but it's involved in many, many reactions. So if something happens to the availability of a coenzyme, it will affect the entire network. And another organizing principle here is that Grant Mitchell, a Canadian doctor, at some point hypothesized that coenzyme A sequestration could be underlying the symptoms in many metabolic diseases where coenzyme A is involved. So if you have a certain substrate, like a fatty acid, that forms a bond with coenzyme A, and that is then further metabolized, what would happen if there is a deficiency in this enzyme that utilizes that further metabolizes this ester of coenzyme A? Well, it would increase this substrate, of course, this ester, But that would, of course, limit then the availability of free coenzyme A. And what he is proposing is that this is a general principle in many diseases where coenzyme A utilizing enzymes are involved. And in many of these diseases, he notices you see these same symptoms like hypoglycemia, also hyperammonemia, and the hypothesis is then that really the limitation in coenzyme A and acetyl-CoA will be the cause of many of these symptoms. So in our case, we were asking the question, is coenzyme A availability playing a role in this deficiency of MCAT? And we use computer models, but we also use cells, we also use mice, and we also use a lot of patient data and samples. So I already briefly introduced the pathway to you, the cyclic nature of the pathway. So we made a computer model of this pathway. And a very important principle again here is that these enzymes, they're promiscuous, because these enzymes, they have to catalyze a number of rounds of fatty acid oxidation. So each of these enzymes can take substrates of different chain lengths, meaning that these substrates are also competing with each other. And that complicates the whole kinetics of the system. So if you would translate this to a rate equation, because we're here in a modeling symposium, if you translate this to a rate equation, then you would see that all these alternative substrates would be competitive inhibitors. So assume that we are looking at the oxidation of C16 by this green enzyme. Then, of course, C16 acyl-CoA is the substrate, but all the different chain lengths that are also accepted by this enzyme, they can bind in the same spot, so they would be competitive inhibitors and decrease the conversion of the C16. And this is then the case for all the enzymes in the pathway. So this gives a lot of complexity. So this is what we try to model. And the models that I'll show you are differential equation models. So we're talking about time derivatives, where the time derivative of each metabolite is the production by all the rates of production by all the enzymes that would produce a certain metabolite, minus the rate of consumption by the enzymes that utilize this certain metabolite. And each of these rates, of course, depends on the concentration of the enzyme that is the catalyst. And it depends on what I here call X, but actually that's a vector of all the metabolites that have an impact on the rate of this enzyme. So that would be the substrates, the products, but also all these competing metabolites. Well, this has been a project since 2012, I think. So the first publication came out in 2013, and then Karen van Eunen published the first model for the fatty acid beta-oxidation in rat, because from rat liver we had most kinetic data available. And she used that also. Well, first of all, she validated this. So she did a lot of experiments. For instance, here you see concentrations of certain metabolites, and you see what she measured and what she modeled. So she did a lot of validation experiments. She also made a model for mouse. And we started investigating different properties of these models. So, for instance, Ventau, a mathematician, he looked at there is bistability in this system. Anne Claire was the first to show the sequestration of cofactors, in her case, in this case, of course, coenzyme A. And what she shows, and that is, of course, already answering the question that I started by, at least theoretically, what she showed is that if you give a lot of substrate, so here you increase the palmitoyl-CoA, so that's the C16 fatty acid CoA, the first substrate of the pathway. If you increase that concentration, what you see is the black line, the free coenzyme A is really decreasing, and you see a huge accumulation of coenzyme A esters, particularly with a chain length of 4 or 6 carbon atoms. And this is all published, but just to give you a flavor of what she showed would be the mechanism behind this. It's not just that this acyl-CoA is accumulating. There is really a vicious cycle that makes it very bad, particularly in this pathway. So what happens is if you give a lot of substrate, then you see, of course, downstream in the pathway, all metabolites increasing. So one of the metabolites that starts increasing is this keto-acyl-CoA at the end of the cycle, because if you give a lot of substrate, then the control shifts to this last enzyme. It becomes the rate-limiting enzyme, or the enzyme with a large flux control for the people working with metabolic control analysis. Well, that in principle is not a big problem, but the problem is that this enzyme, the one before it, has a very, very low equilibrium constant. So if this accumulates a little bit, it starts inhibiting, and then the previous metabolite accumulates hugely, and the previous one, and the previous one. So that's already bad. But the first metabolite here of the cycle is also the last metabolite of the previous cycle, meaning that there will be an inhibition of this enzyme that was already having a lot of control over the flux. So then this accumulation will be worse, and we will start sequestering more and more of the enzyme, of the coenzyme A, which is also a substrate for this last enzyme. So this makes the whole thing totally disastrous. And that's, I think, also, I'll go back to this previous slide, why you do not just see a very gradual decline, but you can see a very steep decline. Somehow you cross a threshold. Well, this is all theory. Here we had Emily Jager, and she is a clinical PhD student, MD PhD student, and she looked back into samples from urine from patients who came into the hospital in a very severe crisis before we had this disease in the neonatal screening. And what she saw in patients, if she had from the same patients urines in a fed state and in a fasted state, and in this first event when they came in a very severe metabolic crisis, then you see that the concentrations of these acyl, in this case she measured acyl carnitines, because those are transported to the blood and to the urine, but it's reflecting hopefully the acyl-CoAs. But what she sees is that it can increase by 10 to 100-fold during this stress. So suggesting that indeed there is a problem with these CoA esters, and particularly the shorter chain, so the C4 and C6. Well, then Christoph Odendahl recently made a human model. So he parameterized the model with all information from human enzymes. He added an enzyme that we were missing so far, and that was the acyl-CoA thioesterase. And it's a very important enzyme, because what this thioesterase does is just hydrolyzing coenzyme A esters. So you would think it's an improductive reaction, because you would like to have this substrate going into the pathway, and this is just taking it out of the pathway. But what it does is that it releases the coenzyme A. So it might, of course, support the process by pulling away the pressure of this huge accumulation of coenzyme A esters. And what he also did to make it more realistic is to partition metabolites between hydrophobic and hydrophilic environments. So in essence, the longer chains are, of course, more in the membrane environment, and the shorter chains are more in the matrix environment of the mitochondria. So he also put that in the model. And here you see again things related between model and these crisis samples from the patients. And here in brown, you see what happens if you only add the human parameters. And in red, you see what happens if you add the human parameters and include this partitioning of metabolites between the hydrophobic and the hydrophilic part. And you see that that really improves a lot the correspondence with the patient data. And you see a lot of accumulation of medium chain acyl-CoAs. Here you see other diseases, just to show that we can use this model not just for one disease, but for multiple diseases. So if there is a deficiency in a very long chain specific enzyme, the VL-cut, then you see the longer chains, C12, C14, C8, and C16 accumulating. And if the deficiency is in the shorter chain enzyme, you see here in blue the shorter chain acyl-CoAs accumulating. And what's interesting is that VL-cut, it's known as a disease that's very severe. because you have already a deficiency in the beginning of the pathway. And this has the strongest effect on the flux. So here the fatty acid oxidation flux is lowest. But the MCAT, the medium chain, the one that we were just discussing, has the strongest effect on the availability of free coenzyme A. So this is worst when it comes to sequestration. And I think it's logical because when you can enter the pathway and go through a number of reactions, then you have a lot of opportunity to sequester the coenzyme A. Well, Christoph also predicted what would happen if you change the enzyme concentrations. So if gene expression in these patients would adapt to the situation. So what happens, for instance, if the short chain acyl-CoA dehydrogenase, if that would increase in activity, then you would see an increase in the fatty acid oxidation flux. And here you see the availability of free coenzyme A that would also increase. And this ACOT, that is the thioesterase, so this is this exit reaction where we had a release of coenzyme A by just hydrolyzing. If you would increase that concentration of the enzyme, you would see an increase in the flux and an increase in the availability of free coenzyme A. Well, the availability of free coenzyme A I can understand because this is a product. The increase in the flux is maybe a bit more surprising, a real systems type of property, because what you would expect, of course, is if you have an extra exit, then for sure you would consume more palmitoyl-CoA. But what we actually see is that we also have more downstream products. So what we plot here is really the production of acetyl-CoA. So meaning that this extra availability of coenzyme A would also speed up these downstream reactions. So it really has a kind of non-intuitive complex effect. And this thioesterase is known as an enzyme that's very important in our metabolism. So it's much more than just a futile cycle. How are we doing with time? We started a little bit late. Do I have a few minutes still?
Yes.
Yes.
Okay. Yes, of course.
Okay, good. I just showed you what would happen if these patients would adapt the concentrations of the enzymes. The question is, of course, are they doing this? Well, we are not taking biopsies regularly from these patients, so we have to do something else. And the simplest thing we can do is use the skin cells, the fibroblasts that we had in store in our genetics department from many decades of diagnosis. So what Emily, again, the same student, did together with Karin Wolters, our expert in proteomics, is that they have been culturing the fibroblasts of the patients under defined conditions, and then they measured the concentrations of the proteins involved in mitochondrial metabolism, particularly, of course, focusing also on the fatty acid oxidation. And here we use targeted proteomics. So it's very quantitative. We just target the proteins that we're interested in. So we use stable-isotope labeled standards for peptides representing each of these proteins. So it's very, very quantitative. So we can look at relatively small changes very accurately. And here you see controls in blue, patients that were symptomatic, patients that were asymptomatic. I always forget what this DND means, but I think it's diagnosis and diet or something. But Emily and Christoph invented this category because they realized that even before the screening, some of these children were diagnosed very early because an older brother or sister had the disease. And then they would be diagnosed early and they would have the surveillance that all these children have now. But there were also truly asymptomatics in this group. For instance, a parent who would be homozygous. You would expect heterozygous parents, but some of them are homozygous. So here you have a homozygous parent. And in her case, the concentration of the enzyme SCAT was extremely elevated compared to all the other groups. So here in green. And also one of the other enzymes, a subunit of the mitochondrial trifunctional protein, is relatively high. Electron transfer protein, ETF, was on the higher end, but particularly the SCAT was the real high one. So the question is, how does this then translate to the flux and the CoA availability in these models? So Christoph put this in his models. And here you see the flux in the different groups and the coenzyme A, the free coenzyme A. And what you see is that indeed flux is decreased in the patients, and so is coenzyme A. And this asymptomatic person is definitely on the higher end. Well, this is rare diseases, so we don't have many patients, so it's not something you can do statistics on, but you can see a principle that these changes in concentrations of proteins, they do have an impact. Only it will be, of course, very difficult to nail this down statistically. So just to finish here, I think we went quite deeply into the mechanisms of the relation between fatty acid oxidation and coenzyme A, and you really see that coenzyme A metabolism is affected at many different levels. You see the sequestration, you see the biosynthesis affected, and a lot of non-intuitive effects in this system. Well, if you translate this, if we can make patient-specific models, we might be able to predict which patient is at risk for developing symptoms. You could also think of coenzyme A as a target for treatment in these diseases. What I personally think is that just giving, let's say, stimulating the synthesis of coenzyme A might not be the best way. Preferably, I think we should pull, not push. So hydrolyze the esters. So for instance, stimulate the last enzyme in the pathway, like the thiolase, or the thioesterases, or stimulate the downstream pathways like the TCA cycle. That's my hypothesis, but that's of course where our future work is directed. We're also currently working with patient-derived induced pluripotent stem cells, because fibroblasts are of course not the most relevant tissue. So we would like to have liver in a test tube, and that's what we have now, but we have no results yet that are interesting enough for you to see. But this is where we stand, and we're also applying these models to other diseases, for instance, insulin resistance and diabetes. That was Marcel's project. So thank you all.